All right, this is going to be chapter 14, Lymphatic System and Immunity, part 2. And we're going to talk about specific defenses, the immune response, types of immunity, properties of immunity, immunology in general, an overview of the immune response, T cells and cellular mediated immunity, a clinical note on AIDS, and then hormones of the immune system. Specific defenses, the immune response. And this is going to be coordinated by the activities of T cells and B cells. And I think you got a pretty good idea that that's the T cells and B cells are different types of lymphocytes. And these cells are going to be specific. T cells are going to be the dumb version of your immune system, which is a cellular mediated response. And then the B cells are going to be more smart. These are going to be designated by an antigen antibody reaction. The B cells also carry long-term immunity, which they'll make antibodies for specific antigens that they've come into contact with throughout their life. Types of immunity. Innate immunity, genetically determined and present at birth. An example of this is if you're a human and there's probably no way that you can get something called ick, and that would be a disease that's carried by a goldfish. So that would be an example of an innate immunity. And then there's acquired immunity, not present at birth and arises by active or passive means. And then that's where these two come into play. Active and passive immunity. Active immunity appears after exposure to an antigen as a consequence of the immune response. So I've come into contact with it, I've made an antibody for it, and now I'm ready for it. Passive immunity produced by the transfer of antibodies to an individual from some other source. Naturally acquired passive immunity, antibodies produced by mother to protect her baby, and these can be passed through mother's milk or through the placenta. So we have resistant immunity, response to threats here. And then we have innate immunity, which is pretty much genetic, right? Humans versus dogs and cats. There are some things that cross, but not everything will cross. And then we have acquired immunity. On the active immunity side, produced by antibodies, and in naturally acquired active immunity develops after you've been exposed to an antigen in the environment. So you had to be actually exposed to the disease or have the disease. An example of this would be chickenpox. That you have the antibodies for chickenpox after you've had it. And then induced active immunity, and this develops after an administration of an antigen to prevent disease. So in this case here, this would be immunizations. And this is going to be, I'm going to have to get the disease. Acquired immunity down here on the passive side, produced by transfer of antibodies from one person to another. Natural passive immunity, and this is going to be across the placenta or in breast milk. This is why breastfeeding is probably a really good thing for babies, because it increases their immune response. Baby's immune system probably doesn't develop until about 2 months out. So the more breast milk that they can actually get, the better chance that they have of fighting off disease. If a infant or a neonate infant has a fever that is less than 2 months, generally speaking, we consider them to probably have something called meningitis. The reason is because the infection just beat back mom's immune system. And then we have induced passive immunity conferred by administration of antibodies to combat infection. And this is going to be a gamma globulin shot or an IgG shot. And these are generally given before you go over in deployment overseas or any time that they would want to enhance or spike the immune system. Properties of immunity. We have specificity. Specific defenses is activated by a specific antigen, and the immune response targets only that particular antigen. Versatility. A person can encounter an enormous number of antigens. Memory. Remembers antigens, immunologic memory cells, remember previous encounters with an antigen and launch faster. Now, this is actually going to make us have a problem later on down the road, especially with anaphylaxis, if we've already been exposed to the antigen. and we've created an antibody. Then the second time we're exposed to it, we can over-exaggerate our response if we're allergic to it. Example of this would be a bee injury and an injected source. Tolerance targets foreign cells and compounds, ignores normal tissues and their antigens. So we allow some certain tolerance in our immune system. Clinical notes, the term immunology, immune response to a complex cascade of events that occur following an activation by an invading substance or pathogen. And a physician who is specialized in the immune system are allergist/immunologist. Clinical notes, immunology, immunity, the ability of the body's defenses to combat infection with specific responses. And we have a primary response. Primary response, in the initial response to an antigen, is called a primary response. Both cells and humoral components of the immune system require several days to respond. General antibodies that are secreted or released are IgG and IgM. Secondary response, a response after being exposed to the antigen for the first time. The immune system responds much faster. Antibodies for a specific antigen are released. That's because I've already seen it, I already have the fix for it, or the special weapon for it, and that's where we'll go from there. Natural immunity or innate immunity. All humans are born with some innate immunity, the ability of the body to protect itself against foreign organisms and toxins. Induce active immunity or artificial immunity, designed to provide protection from exposure to an antigen at some time in the future. An example of this is going to be immunizations. And these are a list of the immunizations. This is from birth to 18 years. And if we'll take a look, hepatitis B1, hepatitis B2, hepatitis B3, what they've done is they have boosted our immune system by giving us boosters through this. Diphtheria, tetanus, get us to where we're nice and familiar with it. After we get into like this age here, about every 5 years we should get a booster. I'm sorry, about every 5 years we should, every 10 years we should get a booster, and then we should get a shot if we've come into contact with any type of thing that would give us tetanus. And tetanus is a toxin produced by Clostridium. Clostridium lives in the ground, so whenever we step on rusty nails and things like that, it becomes troublesome. Haemophilus, polio, inactive version. We do not want the active version. MMR1, MMR2, around 18 or so years old. If we haven't had varicella, to actually get the immunization and then a booster in the future. Pneumococcal virus, hepatitis A series, influenza yearly from about 6 months on. These are adult immunizations, 19 and so on. The years are up here. 1 dose of tetanus diphtheria booster every 10 years. Substitute 1 dose of tetanus diphtheria antipertussis for TD. This is something that we would have gotten in childhood. HPV, 3 doses in females, 0, 2, and 6 months. Measles, mumps, and rubella are the MMR, 1 or 2 doses. Hopefully we have our second dose by 19, 20 years old. Varicella, 2 doses. Influenza, 1 dose annually. Pneumococcal, 1 more dose by the time we get in our retirement years. Hepatitis A, 2 doses, hopefully by 18 months. Hepatitis B, 3 doses by 4 to 6 months. Pneumococcal meningitis, meningococcal meningitis, 1 or more doses. And then herpes zoster, 1 dose. So the immunizations kind of work in waves, and every once in a while, if we don't get refreshed on something, our immune system kind of loses its memory. So if we've had a shot in the past, the easiest way to figure out if we need another shot or not, or another immunization, is to get something called a titer. What a titer does is it allows us to take a look at the antibodies that were made for that specific virus and or disease. With that said, if the titer is sufficient, then they don't need the immunization. If the titer isn't sufficient, then they probably will require the immunization. Immunity. Acquired immunity can either be passive or active. Active immunity, this occurs following exposure to an antigen and results in the production of antibodies to that specific antigen. Passive results in the introduction of antibodies. There are two types of passive immunity. There's natural passive immunity, antibodies crossing across the placenta, barrier from the mother, and there's induced passive immunity, and this is through the administration of antibodies to help an individual fight a particular disease. Example of this is if you contracted hepatitis A, you would probably get an immunoglobulin shot. Clinical notes on immunity, immunizations. Manipulation of the immune system by administering antigens under controlled conditions or providing antibodies that can combat an existing infection. And we do this in a couple different ways. We either inactivate or kill the bacilli, or just remove and have the walls of the bacilli or viral protein coats. Cannot cause a disease, can create a strong immune response, though, which generally may give you some pyrogens and maybe a little fever. Passive immunizations, administration of antibodies. General administration is called a gamma globulin therapy. And then gamma globulin, antibodies obtained from many people, cleaned and processed and injected into other humans. An overview of the immune response. Immune response. Purpose is to destroy pathogens based on the activation of lymphocytes by specific antigens through an antigen-antibody recognition, activation of T cells and then B cells. T cells attack and then activate the B cells. The T cells, or mature B cells, produce antibodies and bind to the attack and bind to and attack the antigen. Okay, we'll get some pictures here in a second and kind of break this down a little bit better. And here kind of we start at them. So these are diseases or bad guys, or antigens. We have specific defenses. These right here, now that they've had an antigen antibody reaction or they've seen these guys don't belong, they essentially send phagocytic cells, which these guys eat, and they send T cells. Once the T cells are activated, these guys start going and they'll attack it. All right, so once the T cells are activated, it initiates this over here, which is antibody-mediated immunity, and we're going to get B cells. And from that, the B cells are going to differentiate into a plasma cell. This plasma cell can create antibodies. When these antibodies interact with these antigens, these phagocytic cells can completely consume them. T cells and cellular mediated immunity. T cells must be activated by exposure to an antigen. T cells recognize antigens when those antigens are bound to a membrane receptor of other cells. Membrane receptors, called major histocompatibility complexes, grouped into two classes, and this is our all-access pass. Class 1 all-access pass, or major histocompatibility complex, are found on the surface of all nucleated cells, and this acts like a red flag. I am abnormal, kill me. Class 2 major histocompatibility complexes found on the membranes of lymphocytes and phagocytes, called antigens. Presenting cell antigens, examples of this are monocytes and our monocyte macrophage group. So these things, if whenever you have this this class 2 compatibility, you have the ability to either differentiate. I am going to eat this guy or I'm not going to eat this guy. Let's talk about one more thing and we'll kind of explain this out. Inactivated T cells have receptors that recognize class 1 and class 2 proteins. So these T cells, even though they're not completely involved in the fight, they're still kind of floating around. They have to be activated. Upon activation, the T cells differentiate into specific functions. We have cytotoxic cells, which are literally what it says, I am toxic to the cell. We have helper cells, which are going to essentially, there's the guy, I saw it all. And then we have suppressor cells. The suppressor T cells kind of put the brakes on the whole T cell immune system cellular mediated response. T cell and cell mediated immunity. Cytotoxic cells are responsible for cellular mediated immunity activated by antigens and undergo cell division and make more cytotoxic cells. So as soon as these guys see that this guy doesn't belong, one, and two, they're having an antigen antibody reaction, they're going to start dividing and making more cytotoxic cells. Helper T cells, two functions. They release various cytokines. They stimulate cellular mediated immunity and antibody immunity. So these things are going to incite the fight. Memory T cells remain in reserve to be able to respond if the same antigen is seen again. And then suppressor T cells dampen the response of the T cells and the B cells. All right, infected cell. And this is going to be a class 1 major histocompatibility complex protein on this thing. So this is your all access pass. This guy is presenting with a protein that says, I'm screwed up, kill me. If this right here is an inactivated cytotoxic cell. So this is in our cellular mediated response. And all of a sudden, because of that class 1 protein on there, he activates. Whenever he activates, we start getting more cytotoxic cells. From that, we also get some memory cells that were also inactive, and these things come into play. This is a blow up of this section right here. We have lymphotoxins that are released. We release cytokines, which stimulate pretty much the eating and the destruction of the cell. And then we have perforin that's released, which will destroy the outside cell membrane. So all through these, all of this stuff, this cell is going to be lysed, or destruction of the target cell. B cells and antibody-mediated immunity. B cell sensitization and activation. We're going to talk about antibody structure, classes of antibodies, antibody function, primary and secondary responses to antigen exposures. B cells and antibody-mediated immunity. The B cells launch a chemical attack on the antigens. They do this by being a part of a series of events that produce appropriate antibodies. The first event in antibody-mediated immunity is sensitization of the B cells. So these guys got to be sensitized to the actual antigen that's delivering. B cells have a variety of antibodies on the surface of their cell. Once they come into contact with an antigen that is associated with that B cell, they become sensitized. Once the helper T becomes activated and binds to the surface of the B cell, the helper T secretes cytokines. This helps mature the B cell into plasma cells and produce more and more and more antibodies. The B cells differentiate into plasma cells and B memory cells. All right, so step 1, we see an antigen. This is an inactive B cell here, and these are antigens. We have a class 2 on this, major histocompatibility complex. The antigens bound to the antibodies here. This B cell is now sensitized. This sensitized B cell will bond with a T helper, which is this right here, the T cell and the B cell together, and from that they'll differentiate. Okay, now they'll cause B cell to divide and make memory cells. They'll activate B cells, which will form into plasma cells, and this is going to produce tons and tons of antibodies. Now these antibodies have the ability to find these antigens and stick to them. This is agglutinate to them or stick to them. What this is going to cause is this is going to cause an antigen-antibody reaction, which will excite the immune system even further. Antibody structure consists of 2 parallel pairs of polypeptide or protein chains, one heavy and one short chain. The structure of the antibody is dependent on the variability of the light and heavy chains. The free tips form antigen binding sites. When the antibody molecule binds to a specific antigen, you have an antigen-antibody complex. As far as the antibodies go, there are 5 classes of antibodies. and our antibodies are immunoglobulins, and these are running around in the actual plasma. If you remember that back from the blood chapter, there is IgG, IgM, IgA, IgE, and IgD. IgG, largest and most diverse. Resistance from bacteria and viruses can cross the placenta and provide passive immunity. IgG. IgM, attack bacteria and are responsible for cross-reaction in blood types. So if we see a hemolytic response in a blood reaction, the antibody that's responsible for that is going to be IgM. IgA, endocrine secretions. Saliva, mucus, attacks pathogens before they can enter the body. IgE, that is bound to antigens, stimulates basophils and mast cells to release chemicals. A mast cell is chock full of histamine and heparin. So whenever a mast cell degranulates, and it looks like it has a bunch of granules in it, they're just everywhere. When a mast cell degranulates, these granules are released and release heparin and histamine into the surrounding tissue. This keeps the bacteria in that location. Instead of allowing him to get back into the vasculature system and run around the body, the vessels dilate, there's a loss of pressure, and everything is thinned out so that we don't get a clumping occurring from the blood clotting process in the body. And then IgD, attached to the B cells and can be involved in their activation. Antibody function, neutralization, can bind to bacteria and viruses, and once it binds to it, it kind of neutralizes it chemically. Agglutination and precipitation. Agglutination, the clumping of complexes together, and the precipitation, when a clump cannot sustain itself in a solution. Activation of complement. This is all what antibodies do. Upon binding, portion of the antibody change shape and expose the areas to the complement. Complement's like a dinner bell. Attraction of phagocytes. Antigens covered with antibodies attract phagocytes. It's like free meal, eat here, eat at Joe's. Enhance of phagocytosis. Make antigens more palatable. So if these guys have an antibody presented on them, they are easily consumed by the phagocytes. Stimulation of inflammation. Promotes inflammation by stimulating basophils and mast cells. And inflammation is going to cause vasodilation and an influx of cells into that area. And this is an antibody. And these are antigens that it is attaching to. This is the antigen here, and then these are the antibodies. And this is what that protein chain actually looks like. These are our binding sites here. Primary and secondary responses to antigen exposure. Primary response, initial exposure to the antigen. Secondary response is more reactive. We've already been exposed to it. We already have antibodies built for it. Secondary response give a large rise to IgG. Antibodies may survive for actually 20 years. So our first exposure, we had a little bit of a spike, and then things went away. The second time, we already had made the antibody and had long-term immunity to this. So now we get an exaggerated response. So the second response is always going to be worse than the first. Clinical note on AIDS, acquired immune deficiency syndrome. A virus entered leukocytes by receptor-mediated endocytosis, attack the helper Ts. All cells affected by the virus die. Suppressor Ts unaffected. The loss of the immune system, decline of the circulating antibodies, opportunistic infections may cause the patient to die. Blood, semen, and vaginal secretions are the way that this is delivered. Clinical signs may not actually appear for 5 or 10 years. Hormones of the immune system. Cytokines are chemical messengers of the immune system. Interleukins. Increased T cell sensitivity to antigens. Stimulate B cell activity and enhance non-specific defenses. Interleukins. Interferons make its neighbors resistant to viral infections, slowing the spread of a virus. Tumor necrosis factors. They slow tumor growth and kill sensitive tumor cells. Phagocytic regulators attract free macrophages to the area. So the phagocytic regulators, these things here are going to incite more phagocytic reaction. And colony stimulating factors. Provide a wide variety of cells, T cells, monocytes, stimulate production of the red blood cells. Patterns of the immune response. Immune disorders, allergies, we're going to talk about anaphylaxis, aging, and the immune system. Patterns of the immune system. The body has specific and non-specific functions, and the figure that I'd like you all to look at is figure 1418 in your book. And we're going to antigen-antibody reactions here. We have an antigen, which triggers non-specific defenses. This is going to give us killer cells and complement any kind of circulating proteins or anything like that, or antibodies that we might have in the area. Will now attack the antigen. And I'm going to erase this here, because we're going to go further down into that. Whenever the antigen was present, we still have specific defenses. The specific defenses activate the T cells, which produce more cytotoxic cells and cause maturation of the T cells. The T helper cells activate the B cells, which look at the memory cells and develop plasma cells, which make antibodies, which fight the antigen. The T cells can now direct their attack. The NK cells and the macrophages can also direct their attack until this guy is gone. This one's a little easier to look at probably. We have a B cell. We have an antigen-antibody reaction with the bacteria. We attach to the T helper. Then the B cell differentiates. One is a plasma cell, which will make more antibodies, and the other one's a memory cell. I will remember you forever. The plasma cells makes more antibodies. The antibodies stick to the bacteria. The macrophage eats the antigen-antibody complex. If we're ever introduced to this guy again, the memory cells will remember that we've seen him before. I remember this one. Red alert, everybody at battle stations. Immune disorders. Autoimmune disorders, immunodeficiency disease, and allergies. Autoimmune disorders develop when the immune system responds mistakenly, targets normal body cells and tissues. An example of this is rheumatoid arthritis, which are autoantibodies which attack connective tissue of the actual joints. Insulin-dependent diabetes mellitus can also be caused from an autoimmune disorder. Autoantibodies, they attack the islets of Langerhans in the pancreas and destroy the beta cells, which secrete insulin. All of this can occur from an autoimmune response. Immunodeficiency disease. Severe combined immunodeficiency disease. Infants cannot produce an immune response, so even a mild infection can prove fatal. And then AIDS, viral infection that targets primary T helper cells. Without the T helper cells, you'll never make antibodies. You'll never get any long-term immunity. And cellular immunity will actually, the cellular portion, the cellular mediated response will actually decline and go down. Allergies, inappropriate or excessive immune response to antigens. Allergens trigger allergic reactions. 4 categories of allergies are recognized. We have an intermediate hypersensitivity, or a type 1, cytotoxic reactions, or type 2, immune complex disorders, which is a type 3, and a delayed hypersensitivity, which is a type 4. Delayed hypersensitivity results from cellular immunity. Does not involve antibodies. Examples of this are going to be a skin rash, and an example of what would give you the skin rash would be poison ivy. And there's a picture of some poison ivy. Erythritol is the oil that's actually on it. It mostly causes this. This would be an example of immediate hypersensitivity, and this is angioedema. Tongue is swollen. This is from a histamine response from the immune system. In the upper airway, this can be problematic because the swelling can close down the upper airway, making it impossible to intubate your patient. Clinical notes on anaphylaxis. Followed an initial exposure to the body creates an antibody. Following an initial exposure to the body, an antibody is created. After a secondary exposure, the immune system is primed. The mast cells contains histamine and basophils, and this responds very, very quickly. Mast cell degranulation releases histamine. H1 receptors causes bronchial constriction, which can give them an asthma attack. And H2 receptors cause peripheral vasodilation, which is going to make them lose blood pressure or become hypotensive. Age in the immune response. With age, T cells become less responsive. Fewer cytotoxic cells respond to an infection. Reduced B cells and antibody levels rise more slowly after exposure. Vaccinations for acute viral diseases are recommended. An example of this is a pneumonia shot and a flu shot every year. The immune system will integrate with every system. Please look over this area. A lot of terms. Acquired immunity. Not present at birth, and this would arise from bypassive means, agglutination or clumping. Allergen, this is going to be something that we have an allergic response to. We've seen it once, so we made an antibody. We have an antibody-antigen reaction. Antibody, examples, antibody-antigen reaction, they're proteins or peptide chains. IgG, IgM, IgD, IgE, IgA. Antigen, foreign invader of some sort. Antigen-antibody complex. The excitatory factor between the antigen-antibody reaction and the immune system. Antigen recognition. B cells and memory cells help with this. B cells is where we make plasma cells, and memory cells and antibodies from. Chemostaxis, or chemical excitation of the surrounding cells. Cytokines is like the dinner bell. Immunity, there's various types. Immunoglobulins, which account for all of our antibodies. Inflammation, which is a cellular mediated response, also can be formed by the release of histamine and heparin from the degranulation of a mast cell. Interferon, again, assisting with the movement of cells towards the area. Monocyte macrophage system, which is going to be a very large macrophagic or eating cell. NK killer cells, which are cellular mediated response. Passive immunity. Passive immunity would be from mom to child. Plasma cells, which are they create antibodies off of the B cells. Pyrogens, which would make fever. T cells, which we have various types. T helper, NK, suppressor, and then cytotoxic. And then thymus, which secrete thymosins, which mature the T cells. And this is the end of part 2. If you have any questions, feel free to call me. Name's Roy Smith, 405-219-7613, or you can email me at smith@imsa.net. Thanks.